Hi there, I'm coming to you from OmnesRes, and today I have a fairly short video for you where we will be covering a small but serious problem you might encounter when analyzing publicly available small RNA-Seq datasets. So what is small RNA-Seq? Well, it's a method for studying small RNAs 20 to 30 nucleotides long, and scientists have to add a 5' and 3' adapter to use this technique, and the machine will read a defined length of the small RNA and 3' adapter. So each read is the same length, but has a different amount of 3' adapter. And this 3' adapter needs to be removed as the first step in small RNA-Seq analysis. So where might you download some small RNA-Seq data? Well, you can go to NCBI's SRA webpage and search for some data. So I have a lot of experience analyzing PAR-CLIP data in 293 cells and the AGO1 protein. I'll probably go over PAR-CLIP data in another video since it's a very powerful technique for studying small RNAs and the proteins they interact with. So once you find a data set you're interested in, you'll get a bunch of information, but no matter where you go, you won't be able to find the 3' adapter sequence. At least most of the time, you won't be able to find it. There's even a GeoAccession you can go to, and that page will have more information, and you can search for the 3' adapter, and they tell you they did in fact use a 3' adapter, and in fact they'll tell you that they removed it when they analyzed their data, but they won't tell you what the sequence is. So these journals require scientists to make their data publicly available, but the scientists don't always give you all the information you need to analyze the data. Okay, so once you found a data set you're interested in, how do you download it? You can just go to that run number in SRA, and they have a method for downloading, but I prefer to just go to the European Nucleotide Archive and paste in the run number there, and then you just get this simple download link. Okay, so how are we going to analyze this data? I'm going to be using Python, so I have a Python text editor on the left and a Python shell on the right. So whenever you are analyzing some data, the first thing you want to do is take a look at it. So we're going to go ahead and open that file, and we're just going to see what it looks like. So for INF, I'm just going to see, what do we have? We have a typical FASTQ format. First, you have the read identity, then you have the sequence, an empty line, and then the quality score. And this repeats. So let's say we want to parse this file and only get the read sequence, which is what we're interested in. So we're going to make an empty list, reads equals empty list. We're going to start with x equals f.readline. Then we're going to do an infinite loop, basically, until x equals an empty string. So basically until the file is finished. So then we're going to append the very next line. So we started with x equaling this, then the very next line is the read that we're interested in, and that's what just got appended. Then we're going to do two f.readlines, and then we're going to set x to equal f.readline one more time. And that's going to just read this entire file. So we start with x equaling this, then we appended this, then we read two lines, and then we set x equal to this again, and that's just going to go on and on and on. So whenever you're reading a very large file, you should make sure that your RAM can handle putting all that data into a list. So we seem to be okay. It's done. We can see how many reads we have. We have over 4 million reads, and we can check to make sure that we parsed it correctly. Oh, so one thing I forgot to do is I forgot to take off the new line character that was on the read. So I'm just going to do a strip right there, and now we're going to be ready to go. So how are we going to find this adapter sequence? I've talked to other scientists, other bioinformaticians, people who have published in prestigious journals such as Science. How do you find an adapter if it's not listed in GEO or SRA? And obviously you can check the publication, check the supplemental information, but it might not be there. And you also may not have access to that journal if you're not working at a research institution. And these scientists, they've told me that it's just not possible to find the adapter if it's not listed in GEO or the publication. So we are going to see if that is the case. So what we're going to do is we're going to take advantage of the fact that sometimes the 3' adapter gets directly ligated to the 5' adapter. And then once the machine runs out of sequence to read, it just starts adding A's at the end. So we're looking for a very abundant sequence, and then we're looking for A's at the end of it. So how are we going to find that? So we're going to make a dictionary. Basically, we're looking for k-mers, and we're just going to go through our reads. So for read in reads, we don't want to do all 4 million because that's a little bit overkill. So let's just do 200,000. And then we're going to take slices of each read because we don't exactly know the length of the k-mer that we're looking for. So we're going to do range 6 to 30. And then we're going to set our dictionary key to be the read up to that position. And we're going to use the get method of dictionaries up to that position, so the same key we just created. If the key does not exist, we're going to return the value of 0, and if it does, we're going to go ahead and add 1 to it. So this is going to take a second to run. Let's see how long that takes. Just to show you what we're doing is we don't know how far to go into the read, so we're taking these slices and we're going up to position 30. If the adapter is longer than position 30, then we'll have to alter our code here. So it looks like we're done. The interesting thing about dictionaries is you cannot sort the dictionary, so we're going to have to convert the dictionary to a list. And what we can do is we can use the zip method to zip the values of the dictionary and the keys of the dictionary. So basically the entire dictionary, the values and the keys, but now it's a list. So now that we have a list, we can use Python sort method, and we can set reverse equal to true, and that will get sorted. And now we can identify the most abundant k-mers in this data set, or at least in the first 200,000 reads. So for I in k mer list print I. So what do we have here? The most abundant 6-mer is this, most abundant 7-mer is that, and it continues to grow, and then we get a poly A at the end of it. In the first 200,000 reads, this showed up 26,000 times, which is a very high percentage, and then it also ended in poly A. So I would be willing to bet that this is our adapter sequence. So we can go back to Google, type in that sequence, and look at that. We get a bunch of hits with adapters. 3' end, 3' adapter. So it's pretty clear that with basically really just this code right here, we were able to find the adapter sequence for this data set. This technique should work for most small RNA-seq datasets. One complication is if you have a very abundant small RNA, such as ribosomal RNA, in your data, then it could be a little bit complicated, a little bit more difficult to find the adapter. I hope you enjoyed this video. The code, this small code, can be found at our website. And if there are any other problems you would like Omnis Res to tackle, let us know in the comments below.